what we're interested in our lab is studying how plants sense temperature. I'd like to outline a few of the experiments we're doing to understand that a little bit more. And this is just my favorite introductory slide. Some of you have probably seen this already before, but it's a nice example of how sensitive plants are to their environmental temperature and how that affects their development and their life cycles. So this is actually a tree that grows in Geneva, and people have very good data sets for when this tree comes into bloom every year. And for the past 200 years, people have measured the day in days from the end of December when this chestnut tree comes into blossom. And for the first 100 years, it was relatively steady. And then, as you can see, in the last 100 years, the time at which the tree comes into blossom has accelerated considerably. So instead of flowering in March or April up here, it's been flowering much, much earlier in the year. And in 2003, it actually flowered in December. So obviously, this is a scientific sample of one, and this is just a single experiment. And we don't know the mechanistic causation, but something that has changed during this time is global temperatures. And in red here, I've just simply plotted the mean change in global temperatures during this time. And global temperatures have increased by about 0.8 of a degree C during this time. So this is a correlation. It's not causation, but it's interesting. And people, of course, have observed many of these similar phenomena in phenologies of plants throughout the world, many different examples. So obviously we can study this in Arabidopsis thaliana, which is a much better system than chestnut trees, obviously for genetics and molecular tools. So these are simply two Arabidopsis plants grown at either 18 degrees C or 23 degrees C. And you can see the plant grown at 23 degrees has a considerably altered development compared to this plant at 18 degrees. So these are genetically identical plants. The only difference is the way they're grown, and that means that temperature must be being sensed by the plant, and that information must be integrated into the plant's development. So this is just a wonderful system for studying the interactions of the environment with genetic programming in plants. And so what we're doing is we're trying to understand the mechanisms of how temperature is sensed, and we want to know how this information is integrated into development. And if we understand these things properly, we should be able to engineer particular thermosensory responses in a controlled way, and that's our sort of ultimate aim of the lab. And so I'd just like to start with a simple observation, but it's quite important. When you grow plants in the cold, they grow more slowly. The development is slowed down. And for a long time, many people suggested that the effect of cold on plant development was simply to slow down metabolism. And the rationale then is that if you grow plants in the cold, of course they flower later, and of course they grow more slowly. And that's simply a consequence of metabolism globally in the plant slowing down. That would suggest there doesn't need to be any particular set of sensory molecules that detect temperature. Simply the enzyme kinetics of all reactions in the plant will slow down uniformly in the plant, and that's why plants in the cold grow more slowly. We don't think that's the case, and one of the nicest suggestions for that not being the case was from a mutant that Nick Harbert made when he was here, which is this knockout for these della repressive genes. So the dellas repress growth, and when you make a knockout of these growth repressors, these plants grow constitutively even at 12 degrees. So a Landsberg plant grown at 12 degrees is very slow growing, it's dark green, and it's very late flowering. But the della mutant grows almost like a plant growing at 22. So if you can knock out just 5 genes and obtain almost normal levels of growth at 22 at 12 degrees, that suggests that there must be regulatory pathways that are sensing temperature and controlling growth and development. And these are really sort of considerably poorly understood. So we took a very naive approach when we started the lab, and I'll go through this quickly because this is sort of published, but we simply wanted to say, can we use molecules in the plant as outputs of a temperature sensing pathway? And to cut a long story short, the nearest we could get to an output for temperature sensing in the plant is a gene called HSP70. And this is simply from microarray experiments. If you take plants at 12 degrees and you shift them to 27 degrees, you see a very nice, robust induction of gene expression for this gene HSP70. And similarly, if you take data which is in the public domain, These were experiments created by Anthony Hall's group in Liverpool. Anthony Hall grew plants at 4 different temperatures, and then he did microarray experiments to ask how gene expression changed at different constant temperatures. And similarly, this gene HSP70, the expression is absolutely proportionate to the temperature the plant is growing at. And this is quite important for us because this gene does not it responds to a temperature increase, but it doesn't require that for its induction. So whatever plant you grow, whatever temperature you grow a plant at, the plant is somehow able to measure that specific temperature and express HSP70 at a particular level. And there are very, very, very few genes in the genome that have this kind of very nice dynamic response to absolute temperature. So the expression of this gene we rationalize is simply the output of some sort of sensory pathway that is sensing the actual temperature of the plant. So we simply took an approach which has been pioneered in Arabidopsis by multiple groups, and as we took the promoter of HSP70 and we fused it to luciferase, and that gives us a great reporter. That's a fantastic tool for isolating mutants that are compromised in their ability to sense temperature properly. And this is the wild-type reporter grown at 22. And you can right away see that this is a 9-centimeter Petri dish, and these are individual seedlings. And this is one of the first mutants we pulled out of this screen, where the plants have a constitutively high temperature response in the plant. And so it's the nice thing about the screen is it's non-destructive. So you can simply do the screen, you can take the seedlings off the plate, you can grow them up and sequence them to find out where the causal mutation is. And one of the first things we found in this screen was a gene called ARP6. And we found that when we had deletions of ARP6, we had a plant that grows constitutively as though it's at a high temperature. So this is our control plant. This is our mutant, which has a high expression of HSP70 luciferase. And this mutant, strikingly, also has all the developmental syndromes which is associated with a plant grown at high temperature, even though this plant is only grown at 22. So this plant grown at 22, it very much resembles a plant grown at 27. So that was quite surprising for us, that we could have a single lesion in the plant that would phenocopy a warm-grown plant. And so when we looked at the transcriptome, we simply saw that actually it was quite remarkable, because all the genes that we see which are upregulated when you go from 12 to 27 in wild-type plants, so this is what we like to think of as sort of the warm temperature transcriptome, all those genes are constitutively on in the mutant. So the mutant has a constitutive expression of the warm temperature genes even at 12 degrees. And this also works for genes that are normally repressed by warmer temperature. So if you plot the genes that are misregulated in ARP610 at a constant temperature against those genes that are induced by temperature in wild type, these are the upregulated genes, and these are the downregulated genes. The upregulated genes, as I just showed you, are constitutively expressed at one temperature in ARP610, and the genes that are downregulated by warm temperature are constitutively downregulated in the mutant. And this is an R-squared of almost 0.5. So you can say that basically this single gene seems to account for about half of the transcriptomic response to warm temperature. And so this is all published, so I'll just summarize with our model for what we think is going on here. So ARP6 encodes a member of a protein complex called SWER1. SWER1 is necessary to insert alternative nucleosomes containing H2AZ histones into chromatin. And so H2AZ nucleosomes are quite special. They have some special properties which alters their biophysical behavior. And H2AZ containing nucleosomes are present at the start of most genes at the transcription start site. They're conserved across all eukaryotes, extremely highly conserved, and they're essential in nearly all systems. We were very lucky in plants that you can disrupt H2AZ function and still obtain live plants. So H2AZ nucleosomes are at the start point of most genes, which is a good place to be if you want to regulate transcription. And they're inserted by the SWER1 complex. So if you knock out this complex, you prevent incorporation of H2AZ nucleosomes. And there are 3 H2AZ genes in plants. And actually, if you knock them all out, you get phenotypes that resemble the ARP6 mutants. So we really think the phenotype of the ARP6 mutant is due to the lack of incorporation of H2AZ nucleosomes in the genome. And somehow, these H2AZ nucleosomes are coordinating the response of the whole temperature transcriptome to changes in temperature. which is quite remarkable. So this is our model, which is sort of at the end of the paper. And what we're simply saying is that these nucleosomes here in orange, containing H2AZ, are really essential to have a correct response to temperature. And the model is that when you grow plants in colder temperatures, a whole set of genes will not be induced. Those genes will be induced by warm temperatures, and that induction of expression at warm temperature is made possible because H2AZ nucleosomes are evicted from the chromatin at higher temperatures. And the eviction of these H2AZ nucleosomes enables these genes to be expressed. And that helps you understand how it's possible that if you knock out or you compromise H2AZ function genetically, then you have a situation which resembles transcriptionally a plant grown constitutively in the warm. Because in the absence of these nucleosomes, if you can achieve this either through increasing temperature, where you increase eviction of the nucleosomes, and we've shown this using a lot of chip experiments. But you can also do this genetically, obviously, with an ARP6 mutant, and then you have constitutive expression of these genes, even at low temperature. Obviously, something's also going on with gene repression, so almost an equal number of genes that are induced when you go from 12 to 27, for instance, are also switched. An equal number of genes are switched off or switched down at least twofold when you go from 12 to 27. And in those cases, we also see eviction of H2AZ nucleosomes is occurring. And the simplest explanation for that is that repressors are able to bind, okay? So this is a working model, and I'm putting it up here because it's very useful for us in our research because it gives us a lot of guidance for the next steps in our experiments. And so if this model is any use, then hopefully it'll help us understand a few examples of plant biology where we've known for a long time that temperature has a big role to play. And I'd like to share with you just a couple of those stories now to show you how this sort of model is helping us understand a couple of questions in plant biology and temperature. So I should also say we also think this is conserved across eukaryotes. So if you actually look in budding yeast, what you see quite strikingly is that there's the correlation between temperature-regulated genes and genes that are misregulated when you compromise H2AZ function is conserved. So budding yeast has exactly the same response as plants. So these genes which are induced by warm temperature in budding yeast are constitutively upregulated in an H2AZ null. And it's the same with these genes down here. They're constitutively silenced. So we think this is likely to be conserved, but we're sort of needs a lot more work. So anyway, our model, and what does it help us with? So let me give you one example of something where we've known for a long time that temperature has a very important role. It's a very fundamental, very simple observation, but it is quite elegant. So this was a really nice paper that came out more than a decade ago. And it's a paper by Bill Gray when he was still a postdoc with Mark Estelle. And what Bill Gray showed was that it's very simple, but if you grow a plant at warmer temperatures, so if you grow an Arabidopsis seedling at 28 degrees, you have a very advanced increase in the length of hypocotyl elongation. So this is a plant grown at the control temperature of 22 degrees, and this is a Columbia seedling grown at 28 degrees. And you can see there's a huge increase in hypocotyl extension. And so this is simply due to temperature. And if you compromise auxin signaling in these plants, so if you use mutants in, say, the AXL1 mutant, you can completely prevent this elongation of the hypocotyl. So this temperature-driven hypocotyl elongation is absolutely auxin-dependent. Okay? And then about 10 years later, Kerry Franklin had a beautiful paper in Current Biology where she showed that actually this elongation of the hypocotyl in response to warm temperature is mediated by a transcription factor called PIF4. Now, I think this is just a really important paper because it establishes this link between temperature and hypocotyl elongation, and it showed that when you knock out PIF4, you completely abolish this increase in high temperature. So we were collaborating with Kerry already, and Kerry was collaborating with Bill. So we said, well, maybe there's a link between this transcription factor and auxin, and what would that be? And so we went and looked for genes that might be involved in auxin biosynthesis and to see if we could see a change in their gene expression in response to temperature. And two of these, TAA1 and CYP79B2, Kerry found were both strongly induced by warmer temperature. So this is at the lower temperature of 20 degrees. If you shift these seedlings up to 28 degrees, you see a big increase in both TAA1 and CYP79B2, which is another enzyme in auxin biosynthesis. Previously, these two enzymes were thought to be involved in separate pathways in auxin biosynthesis, but I think a few months ago it was confirmed that these are actually part of one pathway, which is necessary for the synthesis of auxin. So these two genes are induced strongly by warmer temperature. And what was nice was that when Kerry did the PCR on PIF4, the upregulation of these genes was completely abolished. So the PIF4 is absolutely necessary for the upregulation of these biosynthesis genes. And so at this time, Vinod Kumar in my lab was doing beautiful chip experiments on PIF4 for separate things. But we said, well, if these genes appear to be dependent on PIF4 activity, then maybe PIF4 is binding directly to these auxin biosynthesis genes and inducing expression. And that's what Vinod showed very, very nicely here. So these are just, this is TAA1 and this is CYP79B2. These are the genes that we put on the chip PCR primers here. And we used a set of primers basically spanning the transcription start site of both these genes. And what you see down here is that at 12 degrees, there's lower binding of PIF4 to TA1. And then at 27 degrees, you can see very strong binding at this position 4. So this is very robust increase in binding of PIF4 to both TA1, and you see the same thing with CYP79B2 as well. So PIF4 binds to CYP79B2, and it does this in a temperature-dependent fashion. So that makes a lot of sense because we know that auxin levels increase a lot with temperature. And we know that this process is absolutely dependent on PIF4. And it looks like this is a direct process by which PIF4 is activated at warmer temperature and switches on auxin. And so this is quite a sort of simple linear model, but it's quite useful. And it sort of helps us understand, I think, how higher temperature can basically give you hypocotyl elongation through auxin, and it's mediated by this PIF4 transcription factor. And so just this morning, there was a very nice paper in PlantSci that came out from Peter Quayle's group, where they did very extensive transcriptomic analysis of the behavior of PIF4 and phytochrome responses. And this is a nice paper, and what they show at the conclusion of this paper is that the PIFs have a very large set of genes that they regulate. And you can divide this sort of genes into two classes. One is genes that contain G-box elements that are putative direct targets of PIF4 and the other PIFs. And the other set of genes that are regulated by the PIFs are auxin responsive. And so this would help us understand how you have so many auxin responsive genes which are also regulated indirectly by the PIFs. So that's hypocotyl elongation. So I think it's kind of it's sort of starting to make sense, and that's been a great collaboration, and most of that work was with Kerry, frankly. And our contribution was really doing the chip on the PIF4 showing direct binding. We started with the chestnut tree, and so I'd like to just finish on the chestnut tree. And this goes back to observations that many people have made. The phenology of plants has changed in response to climate change. This paper is already 10 years old. But it was one of the first major papers I saw where people say, my goodness, you know, the timing of plants is already changing. And you can actually measure that. It's quite remarkable. And so the other really interesting thing is that this change in the behavior of plants has also led to changes in the distribution of plants. And they did a very nice study in Harvard where they simply went back to Walden's pond. And Thoreau had taken very, very nice measurements of all the plants growing around in the area of Concord where he was living for a while. And they went back and they checked the records, and they compared the distribution of plants in Thoreau's time with current distributions of plants. And what they found was that there's enormous changes in the frequency and ranges of the plants around Thoreau's pond, and it was non-random. And what they've shown here, this is just a phylogenetic tree. And in red, they've highlighted plants that have decreased enormously. And in blue are plants that have expanded and done very well. And basically, this is not random. So what they found was that a lot of plants, basically, that have gone locally extinct are those plants that actually don't use much thermal information to control their life cycle. So they primarily use day length. And those plants have done very well and have expanded their range and are more abundant than they were 180 years ago. those are actually plants that primarily use thermal information to determine when they flower. That's quite extraordinary. So we've known for, obviously 20 years that photoperiod, which is a major determinant of when Arabidopsis thaliana flowers, is mediated by constants. But what we haven't known, which is also interesting, is the pathway by which warm temperature can activate flowering in Arabidopsis. And so obviously, flowering time is a great paradigm for developmental transition. Temperature and light and lots of other signals are sensed primarily in the leaves. And this information leads to the expression of a gene called FT. FT is a mobile signal that's transported to the apex. At the apex, FT can interact with a transcription factor called FD and activate floral meristem identity genes such as AP1. And a lot of people, della and also Karen Halliday showed very nicely that the thermal acceleration of flowering in Arabidopsis was absolutely dependent on FT. And if you take plants and you warm them up, you have a big increase in FT expression. That corresponds with an acceleration of flowering. And similarly, if you knock out FT, you completely abolish thermal acceleration of flowering. So FT is absolutely essential for the thermal acceleration of flowering, and it's sufficient. And so we were working with Kerry on PIF4 and doing all sorts of other things. So we thought, well, maybe PIF4 is actually necessary for the thermal acceleration of flowering. And that's actually the case. So these are plants grown at 27 degrees C in short day. So wild-type plants grown in short day at 20 degrees, say, are extremely late flowering. They need 30 or 40 leaves before they flower. But as others have already shown, if you grow wild-type Columbia at 27 degrees in short day, you essentially have a plant that flowers as early as a plant grown in long days at 20 degrees. So warm temperature alone is sufficient to compensate for the absence of short days. So temperature is as potent an inducer of flowering as long days is. And that was shown by Detlef a number of years ago. But what is interesting, if you grow a PIF4 mutant, so if you grow the knockout that Kerry gave us for PIF4, you can completely abolish this thermal induction of flowering. So these are PIF4 plants with a knockout, and they have no induction of flowering. So PIF4 is essential for the thermal induction of flowering. And it's also sufficient. So if you overexpress PIF4 under a constitutive promoter, you have plants that are now extremely early flowering. These plants look pretty much like 35SFT. So PIF4 is potent inducer of flowering. And what was very striking for us is that you can take these 35S PIF4 plants, and if you grow them at 12 degrees, you completely suppress the induction of flowering. So this is a control plant that's grown at 12 degrees. You can see it's very late flowering. And this is a 35S PIF4 line, which is identical to this one genetically. The only difference is it's grown at 12 degrees. And now, lo and behold, this plant is almost as late flowering as this one. And so you might say, well, what could be happening is the cold is destabilizing PIF4 protein. So even though you're overexpressing the gene in this transgenic plant, the protein is being destabilized by the cold, so there's no transcription factor around to activate target genes. And so that's not the case in our hands. So these are panels showing plants grown at 12, 17, 22, and 27 degrees. And these are samples taken at the end of night, and then there's a time course from the end of night onwards. And what you can see is this is very characteristic behavior for PIF4. If any of you were familiar with PIF4, it's stabilized in the dark. So at the end of night periods, you have a big abundance of the protein, which you can detect with an anti-HA in this case. And then that protein is degraded subsequently in the light. So PIF4 is unstable in the light. That's already well known. The point is, at 12 degrees at the end of night, you have just as much protein as at all the other temperatures, as far as we're concerned. There are small effects of temperature on the transcript level of the endogenous PIF4 gene, but these are quite small effects, and you still see significant expression of PIF4 at low temperatures. So if there's lots of protein, then what could be going on? And so what we did was we simply wanted to ask if PIF4 could be binding directly to FT. That was one question. We knew that PIF4 was definitely necessary and sufficient to activate FT. And then if this could be temperature dependent. And that turns out to be the case. So we did chip experiments where we asked how much binding there was of PIF4 at the FT promoter. And these are 3 different regions of the FT promoter. You could just focus on this first one, FTC1 anyway. So these are 3 different temperatures at 17, 22, and 27. And what you can see is that as you increase temperature, you see really reproducible and really strong increases in the dynamics of the binding of PIF4 to FT. So this is sort of brings us back to our model. So our model is that temperature changes the behavior of chromatin. This is very important because in cases like genes like FT, where we know that binding increases as a function of temperature, something's happening at higher temperature that enables PIF4 transcription factor to bind and activate FT. So what we're very interested in understanding is the molecular basis of this. So why should PIF4 be able to bind at a higher temperature but not at a lower temperature? That's a sort of open question in the lab. And it relates to our working models for what might be going on with temperature. So you could imagine 2 scenarios. You could imagine temperature is somehow affecting a whole signal transduction pathway that results in chromatin remodeling. So there must be something going on at the level of chromatin because the chromatin at different temperatures behaves differently and has different accessibility to, say, PIF4 transcription factor. So this could be an indirect pathway by which temperature is influencing kinase, say, which influences chromatin remodeling factors, which then causes a change in chromatin, which enables PIF4 to bind, say, in this case, and gene expression to go on. An alternative, simpler explanation would be that chromatin itself changes in response to temperature. This is if the dynamics of the interaction between DNA and chromatin change as a function of temperature, then you could imagine that this would enable genes to be switched on only at the higher temperature. So our data is consistent with both these models, and we're sort of interested in experiments that will help us distinguish between these possibilities. Preliminary evidence suggests that, there do seem to be differences in the behavior. Certainly, H2AZ nucleosomes have different DNA interaction dynamics compared to H2A nucleosomes. So if you look at the accessibility of DNA as measured by the ability to digest DNA which is bound onto nucleosomes with a restriction enzyme, H2A-containing nucleosomes are basically constitutively open. So basically, if you have DNA which is wrapped around a nucleosome, it's rapidly digested when you incubate it with a restriction enzyme. If you take the same nucleosome and which is containing H2A, H2AZ nucleosomes, you incubate it with a restriction enzyme, the digestion rate is much slower, suggesting the DNA is more strongly bound. And that binding seems to vary perhaps slightly as a function of temperature. So at higher temperatures, we sometimes see slightly more accessibility of the DNA, but this has to be repeated. Okay, so one more slide and then I'm done. So what we're doing now is collaborating with a structural biology group to ask whether the rate of DNA, so the unwrapping of the DNA from nucleosomes is altered in H2AZ containing nucleosomes. So this is the structure of a nucleosome with 2 rounds of DNA wrapped around it. These residues in red are the residues that are different between H2AZ nucleosomes and H2A nucleosomes. So we think these are critical for giving chromatin that contains H2AZ this particular temperature responsive behavior. And so we're working with a structural biology group who is making point mutants and analyzing the structure of these residues and where they occur. And what's quite interesting is that they occur on the surface of the nucleosome that contacts a major strand of DNA, which is also what's been shown to affect the rate of wrapping and unwrapping of the DNA on the nucleosome. So I'd just like to thank the wonderful people who did this work. So the temperature screen and all the work on ARP6 and a lot of the work on PIF4, all the chip experiments were done by Vinod Kumar, who was a wonderful postdoc in the group, who's now started his own group here at JIC. Katja did the flowering time work, and Doris Lucician was a postdoc who discovered the effect of PIF4 on flowering time and did a lot of the PIF4 work on flowering time. And the lab's now moved. We're moving this week to Cambridge, and we're just carrying on with the temperature sensing work. Thank you very much for your time.
Thanks very much, Phil. Obvious question. So in an ARP6 mutant, does PIF4 binding to the FT promoter still be temperature dependent?
So in ARP6 mutant, PIF4 binds more strongly. Okay, so that would be...
So it's not temperature dependent?
It's slightly temperature, not nearly as. So we have constitutive binding, which is what we expect because the chromatin is open.
was just wondering if you could comment whether we should be looking for similar mechanism using perhaps different histone proteins in different other temperature ranges and other biological contexts. So I'm thinking, for instance, cold acclimation and higher temperatures for gene regulation, because you're looking at a particular narrow interval. I was just wondering if we should be looking for the same pathways.
Absolutely. So I think that's a great question. I would take a genetics approach and keep a completely open mind. I think this is one component. Certainly in this temperature range it has a big role, but I suspect there's so much other signaling going on. I suspect plants have evolved multiple mechanisms. So I would suggest it's very likely that if you look in the cold, you may see differences in chromatin, but there may also be completely unrelated mechanisms, say channels or anything else. And so I think there are going to be, but I think for me what it's highlighting is that I think these genetic screens have a lot of power, and because they're open-ended. And so I think we just have to keep an open mind. But I would be amazed if there weren't a lot more components that signal in different ways.